in exploring sex differences and acute conditioned morphine withdrawal. So, a bit of background. An estimated 26.8 million people live with opioid use disorder globally, with the highest estimated prevalence observed here in the United States. Over 81,000 deaths occurred as a result of drug overdose in the United States between May 2019 and May 2020. This is the highest recorded number in a 12-month period to date. And it appears that the primary cause of drug overdose deaths during this 12-month period looks to be opioid use. So, it is true that a greater number of men are addicted to opioids compared to women. However, women progress from casual drug use to addiction at a faster rate than men. This is something called telescoping. Behavioral studies have exposed some of the sex differences present in opioid addiction, reward, and dependence. However, studies in sex differences in opioid withdrawal are lacking. So, much of what is currently known about opioid use disorder, and more specifically withdrawal syndrome, is based on data from exclusively men and male samples. And to date, there's been little investigation into the influence of sex on these environmental cues, specifically when paired with drug withdrawal. So, this experiment aims to use conditioned place aversion as a method of exploring the influence of sex on naloxone-induced place conditioning. So the overarching purpose of this experiment is to explore whether sex or estrous cycle differences exist during acute opioid administration and naloxone-precipitated withdrawal by using conditioned place aversion. So our hypotheses were that female rats would be more sensitive to the conditioned aversive effects of naloxone than male rats. And among female rats, aversion intensity would vary across the estrous cycle. So, what is conditioned place aversion? So, CPA or conditioned place aversion is a classical conditioning paradigm used to test the negative motivational effects of objects or experiences. In the case of this experiment, that negative motivational effect would be drug withdrawal. And the apparatus that we use consists of 3 chambers, 2 that are designed for the animal to discriminate between them based on the color and floor texture, and a middle chamber to connect those 2 chambers. And this place conditioning procedure involves 3 phases: the pre-test, conditioning, and post-test. If the withdrawal effect is motivationally significant, the animal will spend less time in the chamber associated with the aversive effects of withdrawal. So the idea here is that when the unconditioned stimulus is aversive, the rat will avoid the compartment that contains cues associated with it. And over the course of the experiments, we took vaginal lavage samples. Here are just a few of the lavage samples that were imaged under the microscope. And essentially, we were able to determine the stage and estrous cycle each animal is in by examining the changes in cell morphology. And this allows us to determine estrogen levels as well as causing the least amount of stress to the animal. So, in terms of the results, these figures show in the pink bars, they represent the time in the naloxone paired compartment, and the blue bars represent the time spent in the saline paired compartment. Male data are solid and female data are checkered. And data are expressed as CPA scores, which was calculated from the difference of time spent in seconds in each compartment during the pre-test and post-test phase. So it was post-test minus pre-test. And although both male and female animals achieved aversion, there was no significant difference between the aversion scores of males and females. The female data were also quite a bit more varied than the male data. Looking at CPA scores across the estrous cycle, we see again there was no significant difference between females with high or low levels of estradiol during the naloxone conditioning phase. However, females with high levels of estradiol during the naloxone conditioning phase displayed significant CPA, while those with low levels of estradiol did not. So based solely on the behavioral data collected, it appears that male and female rats displayed no difference in their sensitivity to the conditioned aversive effects of naloxone-induced withdrawal. Females with high estradiol displayed significant CPA, while those with low levels of estradiol did not. And overall, they appear to display more variability in their responses. So overall, these findings underscore the importance in assessing endogenous ovarian hormones and their effects on mediating responses to drugs of abuse. It's also important to consider convergent sex differences, where males and females may exhibit the same behavior response, but the mechanism underlying that response differs. And this is where our biochemical assays will help us get a sort of a clearer picture in terms of what's happening. And we're currently running some immunohistochemical as well as Western blot analysis on the brain tissue that was collected after the behavioral assays. We're also looking to characterize not only the motivational and affective related measures of withdrawal, but also we're looking at sex differences and somatic components of withdrawal during chronic opioid use. Thank you.